Do you want to know if your food contains GMOs? Genetically modified organisms can't be identified by how they look, taste, feel, or smell. To detect them, you need to be able to sense their genes. Genes are like pieces of code for the design and operation of an organism, passed down from generation to generation. In GM crops, scientists have inserted code from other types of organisms, so that the plants can now do new things, such as survive the application of herbicides, or produce new types of pesticides. But how do you detect a GMO? We want to tell you what you need, what it costs, and how long it can take. Ready? Step 1. Set up your laboratory. Working with GMOs requires a specialised laboratory, and of course, that laboratory needs equipment. In fact, it needs rather a lot of equipment. At the very least, you're going to need a refrigerator, freezer, grinder, ventilation hoods, autoclave, scales, centrifuge, vortex, shaker, incubator, pipettes, test tubes, plates, gloves, masks, computers, and at least one scientist. Step 2. Select your sample. We decided to test the seeds that farmers were about to plant. You could also test a plant's leaves, harvest, or even a processed product. Whatever you choose, you're going to need a sample that's big enough to be representative, but small enough to test. Meaning, if you want to know if a farmer's seeds contain GMOs, it's not very useful to test just one. That seed might be GM-free, but not the others. And at the same time, you can't test every seed because then the farmer would have nothing left to plant. How to get this balance right is a topic of serious scientific debate. Step 3. Prepare your sample. Since genes are sequences of DNA existing deep inside each cell, the first step is to grind the seeds down to very fine particles. Here you need to be very careful. You don't want to spread GMOs while looking for GMOs. If you're testing different samples, you need to clean in between, just to make sure that particles from one sample don't move to another. And any materials or equipment that you use need to be sterilized or burnt afterwards. Step 4. Choose an appropriate method. There are several methods to detect a GMO, so you're going to need to choose the one that's right for you. Some methods are best to test leaves, others for seeds. Some methods search broadly for many types of GMOs, while others search for just one specific type. Some methods give you a rough idea, and others are more precise. Each method has its pros and cons, and this is also a subject of serious scientific debate. We wanted to test seeds for a broad range of GMOs using a method that was as precise and as sensitive as possible. This meant that we chose the method that is known as real-time PCR. For real-time PCR, scientists need what are called primers and probes and a cocktail of chemicals to help them do their job, which is searching for GM sequences and emitting a signal if they're found. There are many different primers, probes and chemistry to choose from, and we chose some recommended by the European Commission because these are supported by many scientists. But this was not the cheapest option. We also still had to test their effectiveness and to adjust the method for our particular sample. Step 5. Extract the DNA from your sample. To extract the DNA, you start by adding a chemical to your ground up seeds to break open the cells. Then you vortex your sample to mix it all together, incubate it, and let it shake so the chemical can do its thing. Once it's done, you centrifuge the tube to spin all the solids to the bottom and then carefully remove the upper liquid layer. Then you add another chemical to break the material down even further, vortex, incubate and shake again. You centrifuge the sample once more, place the upper liquid layer in a new tube with yet another chemical to turn the DNA from a liquid into a solid. To help the cotton-like strands of DNA to condense, you centrifuge your tube until you have a solid pellet at the bottom. Throw away any liquid that's left and clean your pellet of DNA with some ethanol. Air dry it to remove the ethanol, and when this is done, dissolve and dilute the DNA back into water. Whew, that took some work. And just think, if we ran 100 samples, it would be 100 times more work. Now that you've finally extracted your DNA from your sample, it's important to know how much you've got before you start testing if it contains GM sequences. To calculate the amount of DNA extracted, you can use this clever little machine, but it is also really expensive. Step 6. Test the DNA for GM sequences. With the DNA now extracted from your sample, you're ready to test if any of it contains the GM sequence that you're looking for. To do this, you need to add your primers and probes to the DNA, together with yet another master mix of chemicals. You then put all of this into the real-time PCR machine, and it will run through several cycles, raising and lowering the temperature. If it contains the GM sequences you're looking for, the primers will find them and begin to make copies. The probes will also attach and start to emit their fluorescent signal. The strength of the signal will depend on how many copies of the GM sequence you have. Scanning for the fluorescent signal emitted by the probes, the real-time PCR machine sends information to a computer where you can start to see the results. When you test your sample, you need to also test something you know has the GM sequence in it, as well as something you know doesn't. This gives you positive and negative controls and helps you to be sure that your method is working. You should also run the experiment several times to make sure the results are always the same. If they aren't, you'll need to go through the whole process again. Step 7. Interpret the meaning of your results. While you're nearly done, you still need to spend some time doing some calculations, controlling for things like how efficient your primer is and how your sample compares with your controls. Once you've done this, though, your test is complete. Congratulations! Starting from scratch and using this method has cost you approximately $64,000 and taken anywhere from 3 days to 3 months. Of course, if you send your sample to an established laboratory or run additional future samples, it can be cheaper. However, you should remember that your results only apply for your particular sample. For example, these seeds may have been GM-free, but the seeds of a neighbouring farm were not. And cross-pollination could then contaminate your farm later in the season. Or perhaps, seeds you get in the future could be contaminated. Or, you should also remember that even if your results indicated that your samples were GM-free, they may still be contaminated, just not with the GMOs that your method was looking for. Being able to detect whether our agri-food systems contain GMOs is extremely important. However, as we hope to have shown, it's also complex, expensive, time-consuming, and ultimately uncertain. It is therefore crucial to understand how contamination takes place, how to minimize its spread, and insist that those profiting from GMOs take responsibility for controlling them.